Okay, so in this mini lecture, what we're going to talk about is a concept that is very much related to the ideas of development and evolution. It's called homology. I'm pretty sure you've heard of it before. We talk about homologous chromosomes, we talk about homology in a lot of different contexts in biology. So, within evolution and development, and to a certain extent, what we have for the homology with chromosomes as well. What homology means is that it's the same in the way in which it is put together, and it's the same in origin, but it's not necessarily identical. So these developmental processes, patterns, of course, that we'll be studying in quite a bit more detail over the course of this class, and the way in which they define evolutionary relationships are oftentimes defined in terms of homology. So homologous structures are going to refer to structures that have derived from common ancestry. So you remember last time we talked about the development of the limb in the structures of mice, or the way in which the forelimb is built in mice and the bat. So that forelimb, as illustrated by its homology, is going to show us that those homologous structures are related to each other and similar to each other because they came from some ancestral structure, a forelimb in whatever the ancestor was to mice and bats within the mammals. Analogous, on the other hand, which obviously we're not going to talk about a whole lot in this course, but shows up in some of the other biology courses you might study in, is that structures are similar because they perform a similar function, but they don't actually share a common ancestral structure. They don't come from a common feature. So something like a bat wing and a butterfly wing, even though they're analogous in their function, those would not have come from similar structures. Now a bird wing, on the other hand, would definitely be homologous to a bat wing, and of course here would be a seal or a whale limb. We could put in there human arm, a dog leg, a horse's hoof. All of these would be very homologous, and of course what you notice in the homology is the fact that the layout and the pattern and the individual structures are going to be very similar. Now granted, the way in which development proceeds, as we saw yesterday or in that previous lecture with the bat and the mouse limb, is going to be determined by how long a structure might grow and what directions it might grow, so that in some you might have very long digits, in others you might have very short digits, and the way in which these bones relate to each other might change a little bit, but notice how the overall pattern is still homologous. So that kind of homology, as you look at all of the different ways that development proceeds, so the way in which a zygote splits into two cells, the way in which the gastrulation takes place, the way in which different little organs get laid out, is oftentimes going to be used for evolutionary relationships. So here's a picture I actually took out of a 106 book. So you guys remember all of these different kinds of animals and some of the things that you had to learn about each one of them, these different characteristics. So is it a deuterostome? Is it a protostome? Does it have radial symmetry or bilateral symmetry? Does it have a coelom? Is it segmented or jointed? All of these things. Notice how every single one of them refers to some aspect of development, of how that little embryo is going to develop and create the organism. And so development is very much related to the evolutionary relationships that are described in the evolutionary tree. Related to some concepts we've already talked about, let's fold some ideas for evolution in here as well. If you think about what we saw with those mice and bat limbs in that last lecture, embryos themselves across the board tend to share more homology early on, not just in their structure, but also in the processes that they are carrying out, than they would later in development. So as they become more and more specialized, those general structures give rise to those specialized features. So remember, we had the small little limb, in both the mice and the bats, and then over time, as time went by, those fingers got longer in the bat, and that webbing got more pronounced. And basically what happens is in the mouse, that webbing between the fingers kind of disappears. And these would be more specialized features that basically built off of that little limb bud that was very similar in both of those. Here's another example of how things that are related to each other in an evolutionary scale are going to show very similar structures at the embryo level. So shrimp and barnacles are very similar as larvae. And of course these larvae are swimming and self-feeding within the ocean environment. And it's not until later, and in both these cases, they're going to go through several larval or instar stages. And you'll have, in this case, a very different adult from this. This was very mobile, this one stays put. And the important thing to remember is that as these guys slowly over time develop, they become more and more specialized to a specific type, whereas at the embryonic level, they might be built off of the same homologous kinds of structures and use the same molecular toolbox to accomplish those. Remember, our regulatory proteins could be similar in building this eyeball versus whatever light detectors this has, but the bottom line is that those toolboxes, as more and more time goes by, are building slightly different things. Here is a picture of a variety of different embryos, and so you can see they have a lot of similarities here early on, and we'll talk about some of those. And as they grow, of course, here you've got vertebrates, and well, I guess these would be vertebrates as well, but these would be have more yolky egg and extended out, and would be able to self-feed a little bit earlier than others. So at the embryonic level, they are more similar, but as they continue to grow, of course this is still very early in the embryonic development, these would be getting more and more unlike each other. So the further along they go, the more and more unique they get as they take on the material. Well, it's not so much the material, but they take on what their genetics are saying about them. Okay? And related to the fingers and or the bones in the fingers and the arms, here's just another example of it that I wanted to show you. This is the bones in the skull, and you can see how in various different vertebrates here, those bones, even though they are homologous and derived from the same ancestral structures, can in this case take on very different types of functions, and the layout could be slightly different. So for example, this here that's part of the jaw actually becomes part of our ear process, that little those little ear bones, and so on. Here we've got bones that become part of the gills. So homology is really about the ancestry and where these things develop from. Function could be completely different for homologous structures, as we will see. That's a theme that will develop as we compare frogs to chickens to humans and look at their developmental processes. Okay? All right, so next time we will finish off this chapter. I just want to lay out some cellular mechanisms that we will be basically spending the entire first third of this course on in the next lecture, and we'll talk about those then. See ya.